So in lecture, we finished off right in this third out of the 5 steps for virus replication. We had attachment, we have entry, then we have synthesis. And the synthesis for an animal virus can vary a lot. There's different strategies, mostly because of the differences in the genetic material. Now, if the virus has DNA, all replication, all synthesis is going to happen in the nucleus. If a virus has RNA, all of the synthesis parts are going to happen in the cytoplasm. But the biggest issue we really come down to is if it is an RNA virus, because there are some viruses that have a single strand of RNA, but they've got the positive strand, and there are some RNA viruses that are single-stranded, but they've got the negative strand, and then there's some RNA viruses that have both, the positive strand and the negative strand of RNA. And so how they replicate and how they make that new genetic material and the proteins for the protein coat varies. So if a strand of RNA, or if a virus has the positive strand, single-stranded RNA, the ribosome can recognize that positive strand. It can translate it into proteins. It's pretty easy for translation to happen. However, part of synthesis isn't making just proteins, it's also making more genetic material, which means we have to make more copies of the positive strand. Well, cells can't just make more copies of something it sees. It first has to make a template. It needs to see something that it can copy. And so a template is always the opposite of what we need to make. And so if this is the positive strand, we need to make the opposite information, which is the negative strand. We're going to use it as a template, and then we can copy and make lots and lots of copies. If this is an A, a U, and a C, this is going to bring a U, an A, and a G in, because it can recognize those letters and bring in the opposite letter and make lots and lots of copies. And so we're going to get our 2 parts. We're going to get our new genetic material, and we're going to get proteins made. Now, if the virus has the other strand, the negative strand of information, it still has to make copies of itself, still has to make lots of copies of the negative strand, and just like over here, the first thing it has to do is make a template. And so the opposite of a negative strand is the positive strand. So it's going to make a positive copy, may then be able to read it and make lots of copies of the negative strand from it. The only big difference between these is that that translation, making the proteins, comes from the template, because ribosomes can only recognize the positive strand. Now, if a virus is double-stranded RNA, which is extremely unique, only found in viruses, it actually is the best of both worlds. They have both the negative strand and the positive strand, so they have the template for each other already built in, and the translation is going to happen from the positive strand to make all of the proteins that are needed in the capsid. Now, the assembly and release portion. DNA viruses are assembled in the nucleus. RNA viruses are assembled in the cytoplasm. The number of viruses that are made are dependent on 2 things. One, how healthy the host cell is. The healthier the host cell, the more viruses it can produce. And the virus itself. Some viruses naturally produce more viruses in their hijacked host cells. Now for the release. If a virus is an enveloped virus, meaning it steals the phospholipid membrane from the host cell that it infected, it's going to get released by budding. So we can see that happening, that this virus with the genetic material in its capsid is going to bud off of the cell and be released. Now, enveloped viruses are usually associated with persistent infections, partly because when some of these viruses leave the cell by budding, they don't kill the cell, which means our cells stay as permanent virus-producing factories. They're also covered in phospholipid membrane, our own phospholipid membrane, which is a little bit harder to recognize for our T cells. They eventually will recognize that it is a hijacked host cell, but for a while it takes them a little bit longer, so it's a little bit harder to get rid of the virus. Naked viruses, ones that don't steal the phospholipid membrane, they get released by cell lysis. They will burst out of the cell, ultimately killing the cell. Now, some animal viruses can go into a latency phase, which means that the virus can go dormant in our cells. Sometimes it's because they put some of their DNA and become part of our permanent DNA, or sometimes the virus itself is just hanging out inside of the cell it infected, which means it's dormant. Our cells are not producing viruses, they're not being released, the virus itself is dormant. Not all viruses can do that. Some are more prone to do that than others. Viruses that can go dormant are called latent viruses or proviruses, and they can go dormant and hang out inside of our cells for years, if not decades. One example of that are a lot of the herpes viruses. Can do that. Not all, but a lot. Cold sores, that's one of the herpes viruses, that the virus can go dormant for a long period of time, then become active, then it can go dormant again. The virus that causes chickenpox can go dormant, and then years, decades later, it can come back as the shingles disease. Now, your book's got a nice comparison of these stages of replication for the viruses that infect bacteria and viruses that infect animal cells. If you want to take a look at it longer or pause it to look at it, you can. But we're going to talk about how viruses can cause cancer. So all of our DNA has genes, so snippets of DNA that code for something. And we have genes, these snippets of DNA that prevent cell division, so our cells can't divide. We have genes that prevent unlimited division, which means our cells can divide for a while, but not permanently. And we have genes for cell division that we can turn off when we don't want our cells to divide. We have genes that inhibit or stop cell division that we can turn on when we don't want our cells to divide. So we have a lot of checkpoints to stop cells from dividing. That's part of our DNA. However, if something messes with our DNA, we end up with something called a neoplasia, which is new growth. Neo means new, plasia means growth. It means you're going to have cells that are dividing that shouldn't. And they shouldn't because they are incorrect. There is a mutation happening, and they can sometimes divide so quickly that when they are replicating and dividing, the new cells they are making are not correct cells. It's not enough time to put the cells together correctly. And these rapidly reproducing cells form a tumor. And so you can see that these new rapidly reproducing cells don't look normal. They are dividing too quickly. Now, a benign tumor means cells were reproducing very quickly, formed a growth, but then in a benign tumor, the growth stops. Something was able to stop the growth, and the cells stop dividing. A malignant tumor means those cells are still dividing and dividing and dividing. And some of those rapidly dividing cells can break off and end up in the lymphatic system or your bloodstream and travel to new areas, and that's when the cancer spreads. You now have rapidly reproducing cells that are traveling anywhere in the body. Now, how do viruses cause that to happen? Well, again, your normal DNA, you have genes that prevent cell division. It's called a proto-oncogene, the proto meaning inactive or before it's going to actually cause a cell to divide. Because when we're talking about cancer, cell division is bad. We also have repressor genes that repress or stop cells from dividing. And so normal DNA, proto-oncogene, not going to divide. Gene for repressors to stop cell division means our cells aren't going to divide, and you don't have cancer. However, if you get a virus, if that virus has DNA, that DNA can incorporate itself into our DNA. And depending on where it inserts into our DNA, if it inserts somewhere near that proto-oncogene, it could activate the proto-oncogene into an oncogene, which means this is going to cause the cell to divide. Well, luckily, our repressor gene right next to it is still okay. So even though this wants our cell to divide, this is going to stop it from happening. So we still don't have cancer. However, if another virus inserts its DNA somewhere in the middle of the repressor gene, literally just split the repressor gene in half, the repressor gene is not going to be able to do its job anymore. And so that oncogene is going to cause the cell to divide, and you don't have a repressor gene that's going to stop the cell from dividing, so your cells are going to divide. And we don't have anything here to stop it. And so your cells are just going to continue to divide. We don't have the genetic material. That virus DNA is inserted in itself into our DNA at some really critical points so that we can no longer stop cells from dividing. So there are lots of environmental factors that can cause cancer, but a big one are viruses. Other environmental factors, UV light can damage DNA, and if it damages anywhere near those genes that control cell division, it can cause cancer. Any kind of radiation can damage DNA. Carcinogens can cause cancer. These are chemicals that are known to damage DNA. But viruses cause between 20 and 25% of all human cancers. Burkitt's lymphoma and Hodgkin's disease are two cancers that can be caused by the human herpes virus 4. And I'm like, the same virus that can cause mono can actually, depending on the health of your immune system, cause various cancers. Kaposi's sarcoma we'll get to in class, and I'm like, it comes from the human herpes virus 8. And I'm like, that causes cancer in HIV or AIDS patients. And cervical cancer comes from HPV, the human papilloma virus. Now, there are lots of strains of the human papilloma virus, but we know which strain can lead to cervical cancer. And luckily, these days, we have a vaccine for HPV. So we have a vaccine that is now slowing down and preventing cancers. Now, if you wanted to grow your own virus, there are 3 types of media you could use. You could either, 1, grow the organism that the virus needs. So if you're growing a bacteriophage, you could grow the bacteria and then grow the viruses in the bacteria. If you're trying to grow a virus that infects plants, well, you can grow the plants first. If you're trying to grow a virus that infects animals, you could grow the animals in lab, but that's expensive and there's ethical issues with using animals in labs. So they're not usually used all that often. More often, embryonic chicken eggs are used. They are one cell, so one single cell, and they are in their own sterile environment, and it's a really big cell. So you can produce a lot of viruses in that one cell. This is where we get and develop a lot of our vaccines from, is we can grow viruses in the egg cells, but then we can modify those viruses so they don't cause disease, and that's what you get vaccinated with. A third place that we can grow viruses are in cells or tissue cultures. We know that viruses need to get inside of a certain cell, but if we know it needs to get inside of a certain cell in a human or a certain cell in an animal, instead of growing the animal or working with humans, we can just grow very specific cells in a lab. And so that's what this picture is showing. A lot of the cells are grown in these big plastic tubes. They have lots of nutrient media to keep the cells alive, and then we can give the cells a virus to make the virus replicate inside of the cells. Now the question, are viruses alive? Well, the debate is, 1, that they are complex pathogenic chemicals and they're not alive because they don't meet the basic characteristics of life. They can't replicate on their own. They can't do metabolism. They don't have any organelles. They're not a cell. So they're not alive. They don't meet those basic characteristics of life. However, some people, just because of how unique and kind of controlling they are, call them the least complex living entities out there because they do have sophisticated methods to invade cells. They can completely hijack and control their infected host cells, and they can replicate themselves. Not by themselves, but they can replicate themselves. When I refer to viruses, although no, I don't think of them as alive, I do talk about them as alive. So when I say, we're going to kill a virus, you can't kill something that's not alive. So I'm kind of just saying kill means inactivate the virus so that it can't cause disease. But that's a mouthful to say. So I usually just refer to them as a live virus, meaning they're active, or a dead virus, meaning it's inactive, it can't cause disease. Now, the other thing we talk about in this PowerPoint are prions. A prion is an infectious protein, so a protein that can cause disease. Now, it's the main reason why we put it in with this chapter, because it's not alive, it's not a living cell, it is just a protein that's infectious. So in your nervous system, every mammal's nervous system, we have a protein called cellular protein P, so the PRP, and it has all these little alpha helical shapes to the shape of the protein. That's normal protein found in your nervous system. However, some of those helical shapes can straighten out, and they're called what's known as a beta sheet. Now, when you change the shape of a protein, when you denature it, it can't do its job anymore. Now, usually the protein eventually goes away and it doesn't cause any harm. But this particular protein P, when it has changed shape, it's infectious, which means it will cause normal proteins to unravel. It's like a really bad influence. It's going to just kind of tell those, couple alpha helices like, hey, you should really unwind. And they will. And that's where the infectious part gets. Most proteins don't cause denaturation in other proteins, but this one does. And when you have damage to proteins that are found in your nervous system, you're going to have some serious diseases. And they do, because the main disease that's caused by prions is called spongiform encephalopathies. Now, it's a mouthful to say, but the pathy means disease. Encephalo means brain, because when you're thinking of encephalitis, so this is a disease of the brain where the brain looks like a sponge. Because you're going to have death of brain tissue. Your proteins are no longer working anymore. Now, a couple common prion diseases, one is called bovine spongiform encephalopathy, the full name's over here. Where's it? Bovine spongiform encephalopathy, also nicknamed the mad cow disease. Another one is the variant Creutzfeldt-Jakob disease, that's that VCJD. And another one is the chronic wasting disease, which is found in deer in our area. And yes, and I'm like, they have their own prions that the deer, even cows, that can break down neurological tissue. Now, it's transmitted from one person to another by ingestion, so eating infected tissue that has this infected nervous tissue in it, but it can also get transmitted by mucous membranes. That's how they think deer are spreading it, that when they touch noses, because they do, and I'm like, that's one mucous membrane to another mucous membrane. So if you have one infected deer and it touches noses to another deer, it's spreading that infectious prion. And it's hard to destroy. It's not alive. The only way to effectively destroy this prion so that it no longer becomes infectious to normal proteins is incineration. And so that's what they are now doing in our area here in Wisconsin and Minnesota, encouraging deer to take them to landfills where they can get incinerated. Or they're setting up sites where you can take your deer carcasses so that they can get incinerated, so it stops the spread, because we have no cure. So this is showing what normal brain tissue looks like underneath the microscope, and this is what is showing the spongiform encephalo. You've got all these little tiny vacuoles. Now, one of the questions then is, can you get a prion from eating contaminated meat in an animal? Well, there are cases where people picked up the prion from eating infected cows. So what's to say that you can't get it from eating infected venison from deer? There isn't any research that says either way. And I'm like, so maybe you'll see it in the news, but right now there is no data that says either way. Our best right now is we're just trying to test deer to find out if they are infected. You can make your best judgment whether you want to eat deer that are infected. We're trying to prevent the spread of it so there are less deer that are infected, but there's not a lot of research on it because it could take decades before that spongiform appearance appears. And then how do you know if you ate an infected venison sample 10, 30 years ago? And I'm like, we don't have that kind of data yet. So I don't know. We'll see if they come up with anything.